tomorrow about in greater length. Danny worked about a lot of things. So he's going to talk. Abhinav is going to talk about hemoglobin today. Danny is going to talk about lactate. Both are very simple, relatively simpler systems compared to the complex nucleic system that is going to come tomorrow. That is complex tomorrow is drug metabolism, photosynthesis, respiration, physiology, electrophysiology, vision physiology, and all that. All right. So I leave the stage to them. Wonderful guys. Go ahead.
Okay.
No, this is only.
No, this is not mine.
Only.
Yeah. Okay. So good evening. So far, I guess at least some part of murzyme has gotten into you. Murburn concept has, at least a bit of that has gotten inside you. And so far we have been talking with respect to simpler proteins. We have been talking with respect to monomeric proteins so far. And now the system that I'm going to talk about, it's hemoglobin. Now we all know hemoglobin for what purpose? It carries the oxygen, right? Yeah. So we all know hemoglobin for that. It's very famous for that. But then there were certain observations made by our professor, and then we started going behind it. We started to investigate, is this also a murzyme? So let's look into what. Yeah. First, let's check out what is the importance of hemoglobin. First thing, the most abundant globular protein in our body is hemoglobin, and then the clinical importance of hemoglobin also. We check diabetes, and then anemia and thalassemia. All these are very relevant, very widely known diseases that are associated with blood hemoglobin, right? Yeah. And then just now you all said it carries oxygen. It also carries back carbon dioxide. We know that it's a transporter protein. Like proteins have categorization. It is not known as an enzyme. It is known as a transporter protein, right? And then if you remember about oxygen binding, it shows cooperativity. You understand what is cooperativity? What is cooperative binding? Okay, it can accommodate 4 oxygen molecules. The first one, when it binds, it allows the second one to bind even more easily. I mean, the binding of the second one is easier than the first one, and the third is even more easy, and the fourth is all the more easier. So they are all assisting the binding of the next one. That is cooperativity, positive cooperativity. You also have negative, but this over here, this is a classical example of positive cooperativity. And then we know it also does the buffering of blood, so maintenance of pH, etc., hemostasis, right? These are all very well known facts about hemoglobin. Now I just told you there was an observation by a professor wherein this was observed. What is this? ATP, concentration of ATP was seen to be something around 3.5 millimolar. This is clinically established concentration. Okay. So when you see such a high concentration of ATP, 3.5 millimolar is quite high. How does it produce that? You know the structure of hemoglobin? It doesn't have a nucleus, right? It doesn't have a mitochondria. We all know that ATP is generated by the mitochondria. We all know that. So if it doesn't have the mitochondria, where is it coming from? How can it have such a high load of ATP, right? So that okay, this over here is the glycolytic pathway that you have studied straight. Over here you can see there are 2 ATP being consumed, and then there are 2 being released. So eventually what happens, it just cancels out. There is only 0.6 odd micromolar concentration of ATP that can be generated, which is written over here, right? So this is based on the product, the last phosphoenol pyruvate forming the pyruvate, that releasing ATP, giving you some concentration of ATP. Of course, this cannot match the high concentration that is observed, 3.5 millimolar. So that led us into investigating what really would be happening inside here. And obviously, looking at this data, it is very clear that the glycolytic ATP, the production of ATP because of glycolysis, is inadequate. And you also cannot assume that the cells are giving away ATP to the bloodstream, and that is what you're observing, because that is also being consumed inside the cell, right? You need ATP all the time for so many other processes. So you cannot assume such high concentrations being released by the cell inside the bloodstream. So what does it demand? It demands there is something else happening. There's an alternative pathway that must be functioning somewhere. Luckily, we had worked on murburn concept. We had this concept, and with the help of murburn concept, we kind of understood the system, and we have a proposal of how it works. Now, what do we see here? The first thing is you have this methemoglobin, where you have iron in the state 3 plus 3, right? So Fe3 does not allow binding of oxygen. If oxygen has to bind, it has to come to the Fe2 state, right? Only to the Fe2 state oxygen can bind. So this becomes the oxyhemoglobin. Now, this is the general scheme of how it works, and also you have the murburn concept here. You have the draws, which shows that, okay, now one thing that I have missed out just here is this NADH. This was important. I'm sorry, I'll come back to this. Now, we all know that NADH also accounts for the ATP, right? But then, how does this NADH gives you ATP? Yeah, correct, classically. And where does it happen? So can it work here? No, because it doesn't have the mitochondria, right? So this also is negated based on the classical understanding. This cannot function here. So yeah, but if we talk with respect to murburn, you have this reaction that can easily facilitate the reaction that can allow the production of ATP from NADH also, right? You have this draws, you will have ADP and PI, which we are going to talk about much in detail when we are dealing with aerobic respiration. So for now, in simple terms, ADP with an organic phosphate in presence of the draws that will be released here can give us ATP and oxygen, okay? Plus it can be, draws can be, sir has also told us about this, draws can be in the form of superoxide, it can be singlet oxygen, it can be hydroxyl radical, it can be peroxide, it can be any other form of oxygen there, right? Yeah. So this simple thing can explain how ATP can be generated by hemoglobin. Now, that's the theory. Now let's come to the thermodynamics part. Here we have this equation. Now, if you look at this equation, you have NADH, and you have the proton here, you have oxygen, and then you have this ADP, which is attached with the OH, ADP OH, which can give you NAD plus ADP, water, and H2O2. Now, this reaction has a value of minus 546 kilojoules, whose equivalent K equivalent is equal to nearly 10 to the power 96. So that can give you 1 molar concentration of ATP. Okay, so even if 10% of this is working, you can have the concentration of ATP that you have observed, right? Yeah. Okay, this is the reaction basically. This is how the reaction proceeds, where you have this thermodynamically solved equation where we are showing that this is a very feasible form of reaction that happens over there. Okay, so we have seen here at this stage where you have NADH being produced, right? Yeah. So you have NADH that you know that we have yeah.
That also I know how it will be 1 NADH is equal to 2.3 ATP. Those things I know very well.
Yeah.
But here you are mentioning as without mitochondria, how come that ATP is possible?
Because the dross that acts, we are talking about the dross. The dross that acts over here allows this ADPOH to abstract, allow the inorganic phosphate to interact. Yes. Yeah. So it can interact with the inorganic phosphate in presence of dross, and it can give you an ATP. Okay. Okay. Now we'll look into the structural aspects through which we could understand even more depth. Now you have seen here. Yeah. There's a role of 2,3-bisphosphoglycerate over here that we are going to explore and understand how hemoglobin acts as both a transporter plus it also allows the conversion of ADP to ATP, that is ATP generation. So you have this active site over here. You see that this is the heme sitting over here inside here, this place, and this is quite accessible to the solvent. So when I'm saying solvent accessible, that means oxygen can easily go there, right? Yeah. And now you have this 2,3-bisphosphoglycerate, which this is the other side of the protein. This is one side of the protein, and this is the back side of the opposite portion. So this is 2,3-bisphosphoglycerate, which binds at this particular position. Now, from here you can see the channel, right? You can see this channel here. Now, observe this. Over here you have 2,3-bisphosphoglycerate, which is bound to HbA form, and then you have this HbF form, which is the fetal form, okay, where they can carry a lot more oxygen. Okay. Now you have HbA form here, and then you can see this 1,3-bisphosphoglycerate done as a control. 1,3-bisphosphoglycerate doesn't really bind well to the hemoglobin. So what did you observe? You can look at the Kd values, or you can look at the delta G values, which can give you a better understanding. So over here we can see the delta G for the binding of 2,3-bisphosphoglycerate with hemoglobin A gives you a value of minus 10.16 kilocalorie per mole, and with fetal it is minus 8.37 kilocalorie per mole. So it shows really high affinity of oxygen, right? Sorry, 2,3-bisphosphoglycerate. It shows really high affinity of 2,3-bisphosphoglycerate with hemoglobin, but then 1,3-bisphosphoglycerate doesn't really bind well. This is also shown here through in silico docking. Fine. It relates with the data that we have observed. Now, at the beginning itself, I had told you this is a tetrameric protein. So dealing with this with the monomeric form does not give us a very good insight. So now here we are going to look at the tetrameric form, and so it has two units, alpha unit and alpha 2 and beta 2 subunits are there. So there are two alpha and two beta subunits. They sit like this. Okay. Now you can see the cyan color stuff, or turquoise or cyan, whatever you call it. This is the beta, and behind at the back you have the alpha. Okay. Now, in this particular image, we are seeing two different forms. This is the relaxed state, and this is the tense state. Now, in the relaxed state is where the oxygen is bound, and the tense state here you can see this is the tense state. Now we are talking about in the tense state. What do you observe? What is the difference basically? What do you observe as the major difference between the relaxed and the tensed one? Okay. No, sir, it's not the density. If you look at this here, Do you see any gap in the center? No. In the relaxed state, you don't see any gap. There is no channel. But when you look at the tense state, you have a channel. You have a big channel over here, right? Yeah. So this channel is what we propose. It allows the movement of dross. Okay. And when you look at this particular aperture here, this is the place where 2,3-bisphosphoglycerate binds. And when it binds, it actually allows the binding. It allows the oxygen to be held well in the hemoglobin. So when you look at the relaxed state, you can see it's very tightly bound. Now there are 4 oxygen bound. Here you can see the hemoglobin. You can see the heme part here, the red colored. So this is where the oxygen will be bound. But then it doesn't have any aperture, so it cannot be released. It's very tightly. There's no space to move out from there. Okay. So when it is in tetrameric form, there's no leakage as such. It is bound on the heme, Fe, and it can be carried. So it acts as the transporter protein here. Now when you're talking about the tense state, you can see this aperture opening. Now if this happens, what can happen? And we had seen in the previous image, this one, there's this opening. Okay. So if there is an opening, the oxygen can pass through. If it passes through, it can come out from this channel. If it comes out of that channel as dross, because when it leaves the ion, it will be charged, it will be a dross. Now what can happen here? You have ADP bound here. This is the ADP binding site there. Now, we haven't discussed yet. We have done the binding. Bisphosphoglycerate, 2,3-bisphosphoglycerate shows the maximum affinity. Second affinity is of ADP or ATP. Both are almost same. And then comes AMP. So ADP can bind strong enough, but not as strong as 2,3-bisphosphoglycerate. So we can see here ADP can bind here at this stage, and this is at the mouth. So when it comes out, very easily the dross can attack that and allow the formation of ATP, right? From the reaction that we have just seen. Now, yeah. Now this was an observation, not of course this is not our sort of experiment, but xanthine oxidase activity enhances the ATP production in erythrocyte. This was observed in 2006, and now we can understand why. Because it's the dross that is being released, and the dross can enhance the production of ATP. So if you have, when you are doing a lot of exercise, you need more energy, you breathe in more, here it is coming from. Now this is shown in a very simple format here. You cannot make a 3-dimensional direct connotation here, but still for a simpler understanding, you can see that okay, this is the high pH where oxygen is bound. So oxygen binding is facilitated more at high pH, and when you have a lower pH, oxygen binding is not that highly facilitated. So when you have a higher pH, you will have this relaxed state, okay, where you have 2,3-bisphosphoglycerate bound here, and then it's blocking, the channel, and it is not allowing any leakage to happen. Whereas when you're talking, when it goes to the low pH, then what happens? Oxygen binding is not that efficient, and at that time, ATP generation happens. So, and you can see when you have when you have low pH, you have high 2,3-bisphosphoglycerate concentration. So, what does that mean? When you have high 2,3-bisphosphoglycerate concentration, that means you have consumed glucose, and then you need energy more. So, you have to produce more of ATP, right? So, that also correlates with this particular set of experiment that we have done. Yeah. Thank you. Doubts?
Yeah.
when you low and high, can you quantitatively define high and low? What is the figure or something like that?
Low and high?
pH.
pH, I don't remember.
Well, say around 7.2 at high.
7.2 is
High, normal.
normal pH, sir. It's,
So that will
yeah.
be comfortable.
Yeah.
And high maybe about 6.8.
Yes.
Maybe anything between.
High pH. High basic pH. Basic pH.
Low pH.
Low pH is acidic.
Yeah. That
Low,
is about
yeah.
6.8.
6.8. High pH is 7.4. Normal pH, physiological pH that you have. That's high.
But in some therapies, which are other than allopathy, they say the blood becomes lower pH, lower than 6, 7. And many of the disease may occur. They say like that, connected to disease.
When it gets acidic. When it gets acidic. Yeah, that could happen, sir, but then what diseases?
Like fever and everything happens now, that pH goes down.
It's not permanently low, right?
Yeah, no.
Yeah.
Some disease situation, like fever.
In that case, you have permanently low pH. So your body pH itself is going down.
Cancer and other diseases which are permanent, but fever and them are 4-5 days.
Yeah, even for that time. This is dynamic. This is happening dynamically in your body.
Okay. For some time and then it
Yeah.
reaches to that level and then it again goes up. How it manages that pH? Body how manages?
Yeah.
So that theory, your theory is completely dependent on the pH value.
No, not necessarily. We are just linking it with the pH observations that we have reported, not our group itself, but other groups who have reported that low pH has been observed with yes. This work is showing you that 2,3-bisphosphoglycerate binding with the hemoglobin enhances the binding of oxygen, and this is very well correlated here. Plus, when 2,3-bisphosphoglycerate is not able to bind well, I mean when it is getting into the tense state, it's bound, but then it's in the tense state. In the tense state, when you do not have the oxygen well bound over there, then at that time it's also generating ATP. So at one time it is carrying oxygen, at the other time it is also generating oxygen, generating ATP. Yeah. Yeah, it was done. Fluorescent experiment has been done for that. Yeah, we have planned to do it, and we are sure that it can be. I think preliminary studies have been done, but then we want to do it more extensive, and then yeah. More extensive. We want to get inside this more extensively, but then right now only preliminary experimentation has been done with this, and we have seen this happening. Yes, ma'am.
In the pulses and the biological nitrogen fixation process will take place in the root.
Yeah.
In the root, there is a nodule over there, like hemoglobin will be present. Like hemoglobin.
Okay.
Especially because of rhizobium and the plant interaction, like hemoglobin will be present in the nodule. So what I studied with respect to like hemoglobin was only with respect to oxygen. They will hold the oxygen with this, so that the nitrogenase enzyme could able to act over it. They could able to act because the oxygen sends to nitrogenase enzyme.
That's true, but we haven't. I mean, even I have read that, but we haven't checked that system with respect to murburn so far. So I cannot give you a direct comment. you want it could be correlated, but I'm not. I mean, unless we look into it, I cannot make a comment on that.
Okay, sir. So my thing is, as you said, like hemoglobin will under tense condition it will produce ATP.
Yeah, that is for this. But then again, see, this is structure function relationship, madam, and this is not the only system that we have worked with. Only in this particular system we have seen this kind of a semi-stochastic and deterministic approach. So far, otherwise, whatever we have seen, all the other systems, we have seen largely stochastic approach. stochastic reactions happening. So when you are talking about that system, we will have to look at the structure.
No, sir. During when we get tensed, the hemoglobin.
Not that tensed. Not that tensed. Not about stress.
Abiotic stress.
No. We are not talking about abiotic stress. We are not talking about stress. This is the structural conformational change that we are talking about. This is the conformational change. You see the conformational change. That's what I'm saying. Hello.
So what is the difference between this relaxed and the tensed?
The importance that you have heard, it increases your confidence. Understand?
Sir, my question is about the relaxed and the tensed.
Yeah.
What I thought about the relaxed was when you're in relaxed condition.
No.
This is about.
No,
what
this is
exactly.
Yeah, correct.
You should be expressed.
Yeah, even I think when I started, I also thought the same. But then this is not about our relaxed or tense state. This is not about the oxidative stress at all. This is about the structural stress and the relaxed state. This is the conformational differences that you can see here. So this is now stretched, so it is tensed. Now here it is not. It is relaxed.
So when it will take place like that? During which condition?
This happens in the lower pH condition.
So when low pH takes place in the hemoglobin,
that condition will.
Sir, it may be the effect of the tense to pH is lower down. It's not because
It is also when you do not have oxygen bound. That time also it is going to happen.
No, it is the requirement of the body to produce ATP. So that is why mind gives some instruction to change.
We haven't established anything like that, sir. Sir, that is again another speculation. But
I don't understand the pH. pH may be the effect of the
no.
ATP production.
Why can't it be the other way around, sir? Why can't pH can do a lot of things?
Phosphate is being added to the ATP to make it a triphosphate.
Yeah.
So that phosphate is removed from the system, which will change the pH because phosphate is alkaline. Maybe that
Phosphate is not what? Abhinav, go ahead. No, the phosphate alkaline. I don't know why. How can you say something like that?
It's just a thought from my side.
Phosphate is phosphate. Phosphate is an ion.
It's an ion.
No, it will have some pH. It will have some pH in the solution form in the water.
Sir, phosphate. There are 3 pKa values for phosphate.
Once it is removed, the buffering is removed. Maybe
No, sir. We have 3 pKa values for phosphate. So we cannot, and one of them falls something very near to the neutral pH. Other one is in acidic pH, and the third one is in the basic pH.
No,
so
it's a buffering agent.
It is no phosphate. This is inorganic phosphate we are talking about. But then I don't think that is how it is linked.
Change in the structure of the hemoglobin is a natural process. We are not giving any instruction to hemoglobin.
No, we are not. That's what I am saying. It's based on the oxygen that is the amount of oxygen that is present. If there is an excess oxygen load, then it takes up the relaxed state.
So what I feel that whenever the ATP requirement is there.
ATP requirement. When will the ATP requirement be there higher? Usually, when is it higher? When you are getting anoxic. When you are getting anoxic, right?
Physical work.
Yes, sir. That's exactly. So when you are getting anoxic, automatically you will have lesser amount of oxygen. When you have higher amount of oxygen, it is going to be in the relaxed state because there is abundant amount of oxygen inside your body. So you do not need. I mean, of course, the production is being done, but then you do not need urgently very high amounts of oxygen.
Thank you. I think I understand.
As per I have another. So if the in plants, especially in the nodules, that ideal pH will be around the 5.5 to 6.5. So in that condition, whether it will be in the as you said, the low pH will be what will be the ideal pH?
Madam, again I will like to tell you, we haven't worked on that system so far. I cannot give you any comment unless I see the structure. Leguminous plants, sir, like hemoglobin.
Nodules of the plants, especially in the roots, there will be nodules over there. Like hemoglobin will be present. This like hemoglobin helps in oxygen suction so that the nitrogenase enzyme could act over there.
Yes.
This is what I studied in physiology.
All right.
So if as you mentioned about this oxygen consumption, this one. So, as you mentioned, the lower pH will increases the ATP production in the hemoglobin. So, is it having that, whether it will accept to that biological nitrogen fixation process, where the requirement will be more, the ATP requirement will be more.
Now, if you remember, if you were attentive, people would have seen in Lauren Jenkins' talk, did he say something about the proton being involved in ATP? It's not just the ADP PI getting protons. See, this now one very important thing in the understanding, which will be very clear with Danny's talk, is how proton affects the thermodynamics of things. If you see the delta G of formation of H plus, it is around 452 kilojoules per mole. So when you put it at this side, what happens? Will raise delta G. So then see, if anything that consumes proton, will it be favorable or something that produces proton will be favorable? Of course, it is also contingent upon all the other things in the equation. The delta G of formation of all things are important. Now he said that the ATP is produced, the question string arose when he said proton was when in the 10th state, it is at low pH. At low pH, what happens? What is high? H plus is higher, relatively. So what will it do? Sorry, ATP plus? Nobody corrected me.
H.
Alright. So why? Because these two puppies, their pKa, like he said, phosphate has got how many dissociable protons? Phosphoric acid, 3. So all these things, that pKa is very close to that equilibrium. So that is that neutral pH. So H plus, when in the acidic condition, when you have NAD, pH, what does that do? NADPH, what is it a source of? What is it a source of? Electrons. It is not a source of protons. Because if you see the structure, it has got 2 electrons on 1 hydrogen. 1 hydrogen atom. Usually you want for 2 electrons, that 2 hydrogen atoms. This is the reason murburn concept is explaining why NADH, NADPH are involved. Because they have in their electronic structure, they have 2 electrons but only 1 H plus. So now that is a different thing. We'll deal with that in tomorrow's listening. Today, what he started and what she started is the dynamics of H plus ion in the energetics. The important point to be realized is that when you put it on the left hand side, it contributes a large thermodynamic value, which was not realized by biochemists till now. And so what Abhinav's talk till now gave an idea is how till now hemoglobin was seen as a carrier molecule, and what he's saying here is how it optimizes. The next slide, if you put, he has done the docking study, and he shows how the structure that is known, the tense and the relaxed structure, how they correlate with its transporting and oxygen releasing and ATP synthesizing ability. Now, that part is understood. Now you go back to the earlier slide, please, Abhinav. Very interestingly, earlier one. That's the other one. Here, hemoglobin was a tetramer. It is made of 2 alpha, 2 beta. Why 2 beta? No idea. Why should there be 2 beta? No idea. Now we know that hole you see there What is its role?
No, it allows the channel.
That role is purely to make ATP. See, basically,
this is not there in the alpha. This thing, as sir has pointed out, it's only in the beta that you observe this. The alpha, it is alpha 2, beta 2. So only in beta you observe
and
this.
people
in
have
alpha
not
doesn't.
even taken care of this structure. You know Max Perutz? Who is Max Perutz? Ayo. You don't know who's Max Perutz? Yeah. First person to, one of the first structures to be structure elucidated is Max Perutz, and he got his Nobel for elaborating upon the structure of hemoglobin. Alright, so that he worked on cooperativity, and later on we found this is one of the most highly studied proteins. So the active tense form, dense form. Now, what he was elaborating about is the formation, transport, and the structural aspects of hemoglobin, and which is consolidated by what's her name? Ellsworth.
Yes, sir. Ellsworth.
Ellsworth et al.'s multiple paper in which she showed that when you take hemoglobin, this thing is coming on. When you take hemoglobin, red blood cells, you put xanthine oxidase. What is it a source of? Superoxide. So what he showed is hemoglobin, iron 3 and superoxide is exactly equivalent as, in terms of seeing a mixture together, iron 2 and oxygen. Did you understand that part? Fe2 plus. If you don't understand, tell me this. We'll come to your question. We have not yet come to your question. I know that. Fe2 plus and oxygen and Fe3 plus superoxide. Do you understand that they are one? Theoretically, taken together. The number of electrons, oxidation state. If you didn't understand, tell me. No? Yes or no? Don't shake your head if you don't understand. It's simple. See, iron, Fe, so if Fe, it can be ferrous and ferric, two states, Fe3, Fe2. So this plus superoxide, this 2 plus oxygen, this is 1 electron more here, so electronically they are equal. So what he showed is that these are thermodynamic equilibrium and they can dissociate sometimes like this, sometimes like this. When it goes into oxygen, you get oxygen release. When it comes into this one, particularly where it is high pH or low pH, that equilibrium helps. So you need to understand that life, the way you see it, it's a function of thermodynamic state. His question is, brain says to the body and pH release and all that. What we see scientists, we can of course change state. We try to understand a particular state with some condition, another state with another condition. Do you understand what I'm saying? Now, this is a low energy state, that's a high energy state. Tense, relaxed, whatever. And so, in certain, what thermodynamics says is, if this is a state, it has a downhill propensity. If this is a state, it cannot go downhill, and so we need to put some energy into it to get into another state. That's what thermodynamics says. Now, these kinds of interactive equilibrium, and one question she asked, the other slide where we show theoretical. Now, the question here is, it is highly facile. With this, you can make easily ATP. Now, the concentration of ATP that you can get is easily millimolar. So we can explain the concentration of ATP in erythrocytes with the new theory. We cannot explain the structure of hemoglobin with the old theory. beta. Why should we have two
Beta.
betas? Then why he beautifully showed in that 10 structure that ADP beautifully sits. तुमने बोला नहीं ये बात, उनको समझ में नहीं आया. वो ADP sits beautifully at the right place.
ये दिखाया था मैंने यहाँ पर.
You said, but ADP sits there and comes out of that beta monomer, that hole. Where? Show that hole. No, the earlier one, monomer में दिखा. Yeah, that place. Now, why should that hole be there? That is, the dissociation can occur in both ways. So it's a stochastic thing based upon what condition it is. So body's mechanisms kind of, if this goes, अच्छा, proton acidic है, उसको संभालो, energy को minimize करो. Wherever जहाँ से जुगाड़, wherever you can get, you get the things that sustain life by using thermodynamic basic principles. So to ask, now coming to your question, you say that in the leguminous plant there is some kind of leg hemoglobin. Okay, it is some, we have not studied it.
Oh, yeah.
So you say that nitrogen fixing is also done by some kind of redox active enzyme. I can tell you one question is nitrogenase is also surely a murzyme. Nitrogenase is a murzyme. There is no other way it can work. We have not come to that, but it is nitrogen fixing enzyme is the murzyme. I give it to you. So the question that you ask is, how can leg hemoglobin help in that? To understand that question, we have to study that system, and I can tell you for sure at the outset that if something has to happen in a system, in any place, the total certain thermodynamics law processes you have to understand. So in that case, if there is an enzyme that it takes in oxygen or what, like hemoglobin.
Hemoglobin will absorb oxygen and still hold it with it.
Yeah.
It won't release it.
Okay.
So nitrogenase enzyme could activate to do its activity of converting the
Nitrogen fixation.
So that must have to do with
Nitrate बनाता है.
the equilibrium shifting. So I don't know what the system is. We'll have to study that.
Nitrate बनाएगा sir. It will take the nitrogen and fix the atmospheric oxygen. This will work. I am sure.
Yeah.
and then convert into nitrate.
That's what it is. There are multiple nitrogen. There are diazo fixing.
Yeah.
There are nitrogen reducing, nitrogen oxidizing. Or I have not come to that, but surely, if you want to understand the chemistry of it, it has to do with these kinds of equilibrium. And if it is not fixed, it will be a particular state. If it gets fixed, it will be a particular state. And how oxygen dynamics with hemoglobin, if it affects, I don't know. I have not studied this stuff. I know for sure that nitrogen is a murzyme. So it has to do with the equilibrium shifting. That is how it can enable. Otherwise, like what Lauren Jenkins said, you cannot have the same equation giving, you put fruit into a mixie, you get juice and pulp. You put the mixie ulta or rotate it counterclockwise, you cannot get all that. There are states. State. One state, this state is associated with certain things, that state is associated with certain things. Binding of oxygen, relief of oxygen is associated with certain things, certain structures. That's how we have to act. There is order, we are sitting here like this. If there is disorder, sorry, people are going. So that state, it should be maybe sometimes higher energy because there will be bustling. Or depends upon how we see. All these things are very, very crazy, chaotic things. If you come to muscle physiology, you must understand. The classical theory considers that, when we contract the muscle, it is a high energy process. And that requires ATP and all that. But the physicists who argued, Gilbert-Ling and all those people, they said that the contracted state is the low energy state. Why? Because when a person dies, the muscle becomes hard. So you see how we can see the same system with different perspectives. You have to see the whole systemic, all the facts, all the elements of the system to understand where is the uphill, where is the downhill. Not just the delta G, delta H, but also the delta S aspect also. So yeah, the entropy aspect also. So one thing is for sure, her question, I can tell this much, that it is surely a murzyme. We have to understand the system basically because of this kind of equilibrium. I know this answer was a little bit of guma firake, not giving you an answer, but fully sure, absolutely. But at least one part, that it's a redox system. The fixing of nitrogen will necessarily be involved with the oxygen equilibrium, like you said, based on what you data give, but it is through a one electron process, not via two electron process. Because if it is by that simple oxygen binding and release and all that, it cannot explain. Did you understand what I said? Yes, correct.
Plus here, this we have talked with respect to hemoglobin. Same there is myoglobin. Myoglobin cannot do this because if you look at the structure of myoglobin, it doesn't have that. Yeah. So that's why, as sir was saying, you have to look into the structure and then only you can get an idea of what is the function.
And then tetrameric nature of hemoglobin.
Yeah.
Why? Because if it is a monomer, like it's just monomer, what will happen? This is a radical reaction, right? One electron reaction. So if there is another, now Danny will also talk about this. Why do these proteins which are redox active, most often they clump together? Why? Now you know why. Because one electron reaction will generate a one electron thing. So the other monomer will also capitalize on it. So, bhaichara, I give something. So I recruit something and it becomes more profitable, optimized. So they become polymeric and that minimizes the, one electron going here and there. They are locally, minimally. So I have used a word called murzone.
Yeah.
So they minimize the murzone. Thereby, and she was asking me a question, ROS are found in pathological state. So how do you say ROS are good? See, now if there is, I give an example like, now suppose if a engine works beautifully, if there is lubrication, okay? Now, that means that oil is needed for its proper running. Now you imagine oil is clumped and found somewhere, okay, in large amount. Do you say that oil is bad or oil accumulating in one place is bad? Did you understand that? Yes. So something, this reactive species, it is absolutely certain from a thermodynamic, kinetic, all that points of view that it is obligatorily required. Its presence is known, it explains things, but the only thing that we need to understand is that wrong amounts, wrong types, wrong place, that is the problem. Right? If I am standing on my feet, is there anything wrong with me? If I lie down, then there is a problem. Right? That's it.
Thank you.
Now, this paper, his work, the binding concept, which he did elaborate upon it, either acromolar. So yeah, like so basically you see how the data is so beautiful that 2,3 this also here. Yeah, you see the docking, the data that he produced, beautiful 1,3 this also, 2,3 this also. See the changes and the simple in silico methodology. It is not something that we can easily predict. But this is evolutionary training. No, so many years it has gone on. So we can easily get with the binding itself. It's silico methods are not that great. Okay, some of the data you cannot really relay with simple talking. But here we could basically get what do you want? 2,3 bisphosphate should have you get in nanomolar.
Yeah.
1,3 something a little different.
Only a little difference.
Yeah. See
It
that.
goes to millimolar level. Where is nanomolar? Where is millimolar? And it was 6 difference.
And you see here we can get this fetal and the adult. We can explain the propensities of hemoglobin also on these aspects. Not only that, the binding of we did not present the data of how NADP and reduced and oxidized their binding also, how they can once the oxygen goes, this can come and sit there, and it can how they can also help it in keeping the oxygen inside. No?
Locked.
Yeah. So
Locking to यहाँ पर भी we can see this, but then यहाँ से this here we are showing that release बीच से leakage नहीं होगा. ATP generation would not happen. I'm sorry, ATP generation wouldn't happen here. But then when you see that NADH and all, then you can see that oxygen is also locked over there. It cannot just come out so easily.
okay. Next.
Then
Any other question? You're done.
any other questions on this?
Yes.
Yeah, ma'am.
Usually we do blocking to find the targeted site for a particular disease.
Yeah.
So from this inspiration, do you have any clinical significances? Just
No,
basic. Because you have
we do that, but not in this particular case. That kind of study also we have done, but then in here we are actually looking at the binding efficiency and really correlating it with the experimental data that has been already published by so many other groups. So that's what we are showing, that the in silico data of docking is very well correlated with the experimental data that has been already obtained. So that is what is done here. But what you are saying, we do that also.
Because I saw that there were 4 different ligands, like there were small configuration changes in the ligands.
3, I mean, yeah.
In the tabular column which you showed. So I just was inquisitive to know the reason behind having 4 different configurations.
One second.
Just showing that it is effective in distinguishing,
the tabular column which you.
Yeah, I'm trying to get that. Yeah, here, right. This is what you're saying.
Yes, so the ligand part.
Yeah. So here we are showing that this is the tensed and the relaxed state, 2,3-bisphosphoglycerate and ADP. That's all we are showing. Only 2 molecules are there in the relaxed and the tense state. So you can see oxygen bound here, delta G minus 16, and here minus 20. This is the relaxed state. This is more strongly bound here. Okay. So if you talk in terms of KD, which we usually do. Yeah. Okay. I thought delta G is easier for them. Okay, fine. So now you can see here 619 attomolar. Okay, this is the dissociation constant of oxygen in the relaxed state. See here, this is the relaxed state. Okay, this is carbon monoxide. Carbon monoxide binds even more strongly, right? So we can see 2,3-bisphosphoglycerate binds with this strong affinity, and then and that's why getting out carbon monoxide from a bound hemoglobin is much tougher. And then you have oxygen in the tense state. This is again 1.53 picomolar, whereas ADP is 72 micromolar. ADP does not at all bind in the relaxed state. Okay, it doesn't show that. So we have seen that relaxed state. This is the relaxed state.
You see how what is the role of the bisphosphoglycerate? How do the different reaction components, the things that are in your question was, does it have any clinical significance? Anybody? Does it have any clinical significance?
We do molecular docking only for that. So
No. My question is absolutely valid. Great question. I really like that. The point is, can anybody think what could be the clinical significance of this? I think got a published paper in thalassemia reports. It was an invited paper. So, what is thalassemia? There are how many types of thalassemia? Do you know? You know what is thalassemia? Thalassemia is the state in which the genes for it's a genetic disease, right? So there are for alpha.
Beta is not. Beta is deformed in that.
Yes. So the point is, my Guruji's, my professor's daughter, she has thalassemia. Now, the whole approach of everything, treatment or genetic, every approach is only for oxygen carrying and CO2. Yes. The fact that it is a ATP synthesizing thing is not at all considered. So, the roles for the structure, we are explaining the structure of and the structures and functions afresh completely. The states, under what conditions, what is the thermodynamic? So when we try to understand malady, or there are so many other drugs, right? So on some, alright, we are explaining why the common, we are explaining why there is the alpha beta. Earlier, there was no explanation. Why not 4 alpha? Why not 4 beta? Well, so that beta is required. And now, why fetal? Why that binding, that 2,3-DPC, how it acts like a kid in the middle of the tetramer? All that is beautifully structured. See now here, how can for hemoglobin? That I put on the condition. Okay. It's not protein becomes bigger. So here we can have some amount of ATP synthesis in the aqueous state. Why? Because the hemoglobin is jam-packed. Otherwise, what happens is when the superoxide will come out, you have a little superoxide will not. Then, yes, all so jam-packed in the aqueous state. Now, when you see the next phase, next reaction, that is the not tannin thing, the drug metabolism, lipid reaction. They are all aqueous. Chloroperoxidase was aqueous. Hemoglobin is aqueous. Lactate dehydrogenase is aqueous. Tomorrow we are going to do lipid phase, live cross reaction. That is very complex. Simple aqueous system, 1 or 2 things, reactions are supported. When we come to that, yes, so that. Now, thank you, Abhinav, thank you. Molecule doesn't pack properly, and okay.
Okay. So once again, good evening to you. We are going to look at the next system, and I can say that we have some preliminary data to prove that our assumptions are correct in lactate dehydrogenase, thanks to Abhinav's PhD student who has worked on it. She is not here. She was supposed to attend this conference. But with her permission, we are also sharing some of that data. So I want to talk about lactate dehydrogenase, which is again another tetrameric enzyme. Now, this enzyme is very important. You can see that whenever you study about isozymes, you have 5 different variations of LDH. You have LDH 1, 2, 3, 4, 5. Basically, humans have got 4 different LDH genes, which can come from different chromosomes. There is LDH A, B, C, D. Okay, the major ones are LDH A and B. Proportions of LDH A and B come together to form different isozymes. I'll show you how that happens. So we got this paper on LDH published about how lactate dehydrogenase, you know, there is something known as Warburg effect in cancer, where even in the presence of oxygen, there is a lot of lactate production by LDH. So you got lactic acidosis, right? When your blood pH goes down, if your pH goes down, 7.2 to 7.4, we can say is the range. It goes lower than that, we can say that is lactic acidosis. Then we have, what is the role of proton, like what sir was explaining. What is the role of proton in the overall thermodynamic equilibria that is there in LDH? And the most important point we are saying in this paper is that LDH, according to textbooks, is a enzyme which can do both conversion of lactate to pyruvate as well as pyruvate to lactate. Okay, but based on what we are saying, we'll come to that. This enzyme can do pyruvate to lactate very easily, but lactate to pyruvate is almost impossible, which nobody has said before on earth. We would be the first people who would say that LDH is not a reversible enzyme. I don't know whether you know about it, might come as a shock to you that LDH is not a reversible enzyme. So this is the first question that we asked is, you see that there are different types of LDH, 1, 2, 3, 4, 5, as I told you. You can have 4 monomers of LDH A coming together. Okay, LDH A is what is present in the muscle and liver. LDH B is what is present in heart tissue, for example, predominantly. So we can also say M4 or H4. In between these, you can have mixtures of, like if you start from H4, heart, H stands for heart, M stands for muscle. Okay, easy convention. If you have mixtures of these, you can also have M3H or HM3, right? H2M2, H, right? Like that you can have H3M, and you can also have H4. So one thing you should keep in mind is that the predominant form of LDH that is found in muscle as well as liver is your LDH5 or M4, which comes from LDHA gene. I don't know. Okay. So LDHA gene, you can see, we asked this question. There is about 4 times more LDH in the muscle than in the liver. But there is something called Cory's cycle in biochemistry textbooks, where when, let's say, there is an athlete, this person runs a sprint. At the end of the sprint, what happens? All the oxygen that is there in the muscle tissue gets consumed, and then there is fermentation, right? Anaerobic glycolysis, anaerobic respiration, basically. So there is, whatever pyruvate is there is going to be converted into lactate in the absence of oxygen or hypoxic conditions. But there is 4 times more LDH in muscle than in the liver. But then, according to Cory's cycle, what happens is there is glucose, which leads to formation of lactate in the muscle, and then later what happens is that lactate, a minuscule amount of it can go into the mitochondria, but that is not very, very feasible maybe. Most of it will be transported into the blood, where you have another form, predominant form is LDH2. Then it goes into the liver, lactate is reconverted, and through gluconeogenesis, glucose is formed, and again glucose comes into the blood. This is basically what is Cory's cycle. So, first question is, why is lactate even transported to the liver? Because according to biochemistry textbooks, this reaction is reversible. So it should be reconverted to pyruvate in the muscle itself. So when we discussed this over the phone, then we started exploring this, sir, I and also Abhinav. We started exploring how or why, LDH is, how it is involved, what is the role of oxygen in that, why this Cory's cycle should occur. Because there's more in the muscle than in the liver, just remember that. So another point is lactate metabolized in the mitochondria? So one point I should tell you here is that the H-type LDH, they say that it converts lactate into pyruvate better, hot. They say it is aerobic type LDH. And the other one they say is the anaerobic LDH, which is your liver or muscle LDH. So we wanted to explore if there are any differences in these proteins, in their structures, in their ability to bind pyruvate and lactate. So also one more question, why is lactate produced in the presence of oxygen as well in Cory's cycle, and what is the relevance of LDH in that? So if you see your entire glycolytic pathway, many people now agree that the glycolysis does not end with step number 10 where you have pyruvate formation, because the concentration of lactate is to pyruvate is like 10:1 in tissues. Okay, so the last step of glycolysis would be lactate formation. That is what many people say. So another thing is that if you look at this pathway, you got all these stars. Those are the negative charges on the molecules. You can also see the energy values that are there in kilojoules per mole for all of these steps. There is a key step here where you have step number 6 where NADH is formed, and you also have H plus there. But you also have at the end you have lactate being formed, and you can say that you got lactate. Look at the concentration of lactate that you have is about 1.6 to 16 millimolar. When you compare pyruvate, which is much lesser. So you can one point there, which is yeah.
It's not working.
The zoom.
What is zoom there?
No, zoom lesser.
Zoom lesser.
So you got lactic acid. They say that more lactic acid is there, then it is acidosis. But the pyruvate is much more acidic than lactate based on the pK value. So we'll go to the next slide. Okay, this is what you have. LDH itself, the isozymes of LDH and the concentration of LDH, lactate pyruvate ratio, LP ratio, is known to be, a diagnostic marker for several diseases, like even mitochondrial disorders. No, leave it, Abhinav. So if we go back to this slide, you can see that there is a proton being released here, right? So what happens is proton consumption also occurs in the LDH step. So lactic acidosis technically should not occur, because whatever proton is produced here can be consumed by LDH also, and therefore it can be like a proton stat, like pH stat. So therefore the pH should not change or it should not decrease. But then that is what they say, there is a term lactic acidosis. But then we should remember that in all these steps, one main thing is you got lactate at the end, not just pyruvate. There is way more lactate than pyruvate. Now we come to how this enzyme is. You can see that it is, as a monomer, you got the LDHA, LDHB, both of them look similar. And they form these, you can see a very nice groove here, like a cave or whatever it is, where NAD or NADH, they show in this crystal structure that it is bound. And there is a 10 angstrom deep well like that, inside this part. What you are seeing here is the nicotinamide adenine dinucleotide coenzyme, but you also got a cleft there where they say that pyruvate and lactate go and bind. Now you must understand, in what they are saying conventionally is that NADH binds first, the coenzyme binds first. So somehow your lactate or pyruvate has to wade through, NADH is already there, right? It has to wade through, cross that and go and bind. So then we ask the question, are the active sites of A and B different? Because people are saying that A and B, both of them are, one is aerobic, other one is anaerobic. One has preference for lactate, that is heart wala one has got preference for lactate. The muscle isozyme has got preference for pyruvate. Is there any difference? You can see that practically the active site, these are crystal structures by the way, so you got two crystal structures, both of them, left side you got LDHA and the right side you got LDHB. You can see that there is oxamate bound there. Oxamic acid or oxamate is known to be an inhibitor of LDH. And you can see that there are two arginines, R105 and 168. They say that the positively charged arginine attracts the negatively charged alpha hydroxy acid moieties. That is, you got lactate as well as pyruvate, which are negatively charged. So they get attracted and they bind through these arginine. And there is a conserved histidine there, which plays the role of a acid-base catalyst. Histidine can be protonated as well as deprotonated because its pK is closer to the physiological pH, so it can give as well as take protons from substrates that are bound to it. That is the classical theory. So if you compare conservation and all that, I don't want to go deep into these things. I've already showed that. So this is the thing that Sir was talking about, where you got the nicotinamide and there is a change of 1 H and there are 2 electrons which are transferred. So according to Linenger textbook, the conversion of pyruvate to lactate has got a delta G of about minus 25.1 kilojoule per mole. So if you reverse it, it is plus 25, right, for lactate to become pyruvate. That is very easily surmountable in terms of thermodynamic reversibility. That is very reversible. So if you see the mechanism of the enzyme, there is histidine 195, right, which has got the proton, and that proton can be donated and all that. That is acid-base catalysis, which you have. So if you see, we have got two things that people say can happen. One is the forward reaction and the other one is the reverse reaction. We term forward reaction as lactate to pyruvate because the name of the enzyme is lactate dehydrogenase, right? The reverse reaction would be pyruvate to lactate. So you can see that we have put all the values of Kd and Km of the enzyme. Now we can say that the active sites are identical, right? I've shown you that slide. Active sites are identical. NADH binds first and then only lactate, and I told you that going through, when there is a large molecule like that, these small ones have to, squeeze through perhaps, because that entry channel is very confined. It's narrow. It's 10 angstrom deep, but it's narrow. And the forward and backward reactions are actually reversible according to literature, right? But they made a huge mistake in their energetics of the reaction, where, if you take the textbook values that you have, there are many ways to calculate delta G. One is through minus NF dot into delta E, you get minus 26 kilojoule per mole. You can also take the equilibrium constant and you can calculate, you get about minus 66. Or you can also you can calculate the delta G of reaction by taking the sum of the delta G of products and minus the delta G of the reactants. So if you see the textbook values of the redox potentials that are given, the literature values, you will end up with these values if you calculate. But what they have ignored is the contribution of H plus, and you can see that when you add this term, right, you get a energy of minus 476.4 kilojoule per mole, and the equilibrium constant is roughly around 10 to the power of 83 per molar. That is, it is always going to go from pyruvate to lactate. If you got a equilibrium constant of 1, then you can say that it can go this way and that way. But when the equilibrium constant becomes higher, the reaction is going one way. So here it is going from pyruvate to lactate, right? You can see very easily that this pyruvate to lactate here has minus 476. But if you reverse that, if you want to make lactate to pyruvate, it becomes plus 476, which you cannot easily cross. That energy barrier is very high. So we kind of guessed that pyruvate to lactate is very feasible because the equilibrium constant is very high, but lactate to pyruvate, because if you reverse the sign, plus 476, it becomes very difficult, and to get that much of energy, you need something to support that, to reverse it, right? So this is a energetically disfavored forward reaction. By forward reaction we mean lactate to pyruvate. So sir also studied other reactions that are happening, for example with other alpha hydroxy acids. You can see not only lactate and pyruvate, there is also the reduced and oxidized forms of glycolate, glyoxylate, malate, oxaloacetate, isocitrate, and all that. You can see that almost all of them, you got that energy being very, very high, right? So here we also came to this conclusion that there would be some role of ROS. In literature, there is also peroxide formation in LDH reaction, and there is also the involvement of other ROS which are there in the reaction. Now we have to ask two important questions. Lactate, which is generated in the muscle, there is also something known as a lactate transporter, and the lactate tends to go into mitochondria. Number two, lactate goes also to the Cori cycle to the liver. Now classically both these places are very rich places where you have a lot of ROS production, right? In mitochondria there is a lot of reactive oxygen species. In liver, because you have a lot of cytochrome P450 enzymes, liver is very reddish brown in color because of the presence of cytochrome P450 enzymes, and heme is the coenzyme that is present in those cytochrome P450s. So why does it go to the liver? We got to answer that question, right? So if you take RH metabolism, RH is any drug, any generic drug we can say is RH. So the role of cytochrome P450 would be to insert one oxygen into the drug. Okay, so RH becomes ROH. Okay, I hope you are able to understand this. So in liver microsomal cytochrome P450 metabolism, you got a drug RH, it can become ROH. So there is consumption of NADPH by the liver, right? And then you got NADP plus and OH minus being formed. Now if your lactate reacts with NAD plus to form pyruvate and NADPH plus H plus, and if you write how this RH metabolism, drug metabolism, along with drug metabolism, if lactate also goes there, because of the presence of ROS in the liver, lactate can again be converted to pyruvate. So here you got your drug RH, you got lactate, you got NADPH, and you got O2. Now your O2 becomes H2O, and you also got ROH coming from RH, and lactate has become pyruvate. But simultaneously NADPH, NAD plus can become NADP plus and NADH. So you got even NADH formation, right? So when LDH works from lactate to pyruvate, what happens? Lactate is a reduced molecule, right? Pyruvate has got only CH3CO, CO minus. Lactate is CH3, CHOH, CO minus. So lactate is a more reduced molecule than pyruvate. Do you agree or not? So the protons which are there in lactate, they can go into formation of something else. right? Even hydrogen peroxide can be formed. So if you see this overall reaction here, where what is the role of oxygen here, right? So along with your P450 metabolism, when your P450 is working on a drug, there if your lactate is there, you can also, lactate can get converted to pyruvate. So now whatever you have, H plus and OH minus that you have, that gets fused to form H2O. So this mechanism you can see is able to, it is energetically favorable. We'll show you that even peroxide formation, you can see that peroxide can be formed from LDH reaction, that is reported in a paper. So if we come to what is the role of this, I think I'll do this later. So here you got, we also thought, let's check what is the role of substrate binding. Okay, substrate binding, like lactate and pyruvate, where does it bind? What about inhibitors of lactate dehydrogenase? There are lots of inhibitors that people use. They want to take lactate dehydrogenase, they want to put inhibitors of LDH and stop LDH from working, and that is a way, you know, in cancer therapeutics. So how can these lactate dehydrogenase system be inhibited? So we found in literature that there are many inhibitors that people have worked on. You got so many inhibitors which are natural molecules like gossypol and many other flavonoids and all that, which are too large, I can say, to enter the active site of LDH. Okay, they are too large. The active site is quite narrow. You already have NADH. So there are different ways that the enzyme can be inhibited. We can say that there are certain things like pyruvate or lactate mimics, like we saw the example. You can also have crypt blockers. Something goes and binds to the entry channel and stays there, and so there can be no access by NADH or the substrate. There can also be interfacial inhibitors. Now they are looking at how you can disrupt the LDH tetramer and you know, prevent LDH from acting effectively. So there are interfacial inhibitors, but what we are saying is that all these inhibitors, most of them are very good antioxidants. So, and we did docking of several inhibitors as well as substrates. And this is just an overview.
Can I, with your permission,
Yes.
I just break and ask the students if they understood what you just presented in the earlier slide?
Which one?
That earlier slide, not this one.
This one.
Not this one. LDH, triangle.
In this.
Yes, people. What is happening is, if you had the last session, I said something about enzyme. What is this violating? What is the textbook idea giving wrong, very outrightly wrong idea? What is being advocated? Now, see, the point is, I want you to think independent. You're spending your time. This is a good time. Pleasant, you could always be outside, but you chose to come here. Certain things, we are trying to get you to think beyond the textbooks. So Danny now presented something very clearly, what is given in textbooks, what is given in papers. How do you think this is wrong? Outrightly, anybody should have said this is wrong. How come they would ever say something like this? Is it okay, Danny? stealing your thunder, but
No, it's okay.
yeah. What could be wrong? So LDHA and LDHB, you already said, the active sites look very similar. Everything is very similar. One takes from pyruvate to lactate, yes or no? That's what it says, no? Yes. Why should it be wrong? What is the definition of an enzyme? We discussed that they have gone for it. Please, let's just. Yes. Confidence, blunder, pushpe, confident. Yeah. For microphone, please. She should learn to speak.
It's there.
No, there is nothing grinning. Don't grin. That's silly. Nothing. No grinning. Speak freely. Speak confidently. I'm saying this is the purpose. You understand? No? Yeah, but don't get excited. Don't get excited. Then what happens is when you grin and laugh and smile, you're messing with your scientific temper. Something that you're doing seriously, you would rather do seriously. We want to have fun, we have fun. Alright? Yes. Yes. I said something about equilibrium, thermodynamic equilibrium. What does the enzyme do with respect to the positions of the equilibrium? That was the question. What's your name? Aruna. Everybody should hear you.
Hello, நல்லதானே வருது?
Enzyme is a biological catalyst.
Yeah, you said that. But what is the other part? We discussed. Guys, here, anybody? Remember? It only allows attainment of equilibrium. Yes, exactly. That's very important. So, what does the equilibrium dictate? Yes, good. You said something? That's partly right, but that's not what was the crux of what we are discussing. So, what is so awkward about this proposal? Now, when you put what does the equilibrium tell you? Basically, the position, the data. Now, if you, in normal state, would you imagine that, see, I can jump from here to here. I can jump from there to here. But, from here to Everest, can I jump that way? From there I can come down. I mean, happily true. But, theoretically, it is possible, right? That is what these values are supposed to do. So now, here is something that I'll very clearly say. I wanted to interrupt because the attention span goes off after some time, right? So that's why a lot of details, they're already, it's AC, after some time their mind starts wandering. Now, if I say, why should something be feasible or facile or possible while something is not? Enzymes have no brain. Alright? They just do certain things. I mean, there could be, I don't know. We don't know. We are only at particular level. Sorry, I take back. Not that intelligent, but we know how they work generally. So basically, like we said, if an equilibrium condition is that it will be 5, 1, 2, 3, something like that. That will be the general condition. The delta G is calculated at what conditions? Normal temperature, normal temperature, ideal concentrate, but 1 normal, 1 molar, all that. No, the standard D. So when you give a delta G, it kind of says this state, that state. You get an idea. Now, what does this guide? What do your biology textbooks advocate with this kind of a proposal? Why did they do that? Now, here slowly you will start asking why, how, and all. So now here you see, they see that LDHB is in where? Which place?
Heart.
Where? Heart. Who said heart? Somebody said heart. Yeah, that's right. Heart is the place. So does heart have lactate? See, now where do you get lactate accumulated? Muscle. Quick stress, that's sore feeling. Heart keeps on pumping, never has cramps, no lactate deposition. So they found that LDH3 is in heart. So it must naturally, suppose, now I'll give you an example. They found some elephants. This is the most favorite example. They found some elephants in Mansarovar, near Mansarovar in Tibet. Okay, and the elephants had Appukutan, Kunyiraman, Velayadan, some nice Mallu name tags. They look like Mallu elephants. Okay, so now there were 4 of them, and sometimes when they find 4 elephants there like that, they also found a Maruti 800 with Kerala registration. Okay, so somebody put forward a theory. So these 4, and sometimes it rained. So some theory like that. When it rains, some Mallu elephants from Kerala take a Maruti and drive down from Kerala. So there should be some line and logic. 4 elephants, 4 cars, it's 1-to-1 correlation. Wonderful. So something fit, observation fit. Then Kerala, tag, all. But then something should irritate. Yes. What are the things that irritate you? How elephants fit in. Wonderful. Why elephants are large, car is small, and also I could not say the time factor. Sometimes elephants were seen in Kerala, say Monday afternoon, and they are in Tibet Monday evening. So even if they are going by a car, highly unlikely that they will reach. So even if they can go, fit in the car and reach in 2 hours, it's highly unlikely that they can drive. Even if they can drive all the way until there is no road. From Kerala, direct road to. There are multiple level of problems. Just because the heart doesn't have lactic acidosis or cramps and all that keeps pumping, just because there is lactate dehydrogenase B there, it's not because it can violate the principles of physics. Did you understand? But you cannot, that kind of a thing is what has happened. Now these 4 elephants and 4 seats in Maruti car will be useful for you when we'll talk about that most important question: 4 electrons, 4 protons, 1 molecule of oxygen making 2 molecules of water, all that, that electron transport chain. But the idea that these things, the elephants, 4 elephants. Now you people are doing bio stuff, right? Now I say a civil engineer, if you ask him, how big is this room? Okay, this is 20 feet by 15 feet. So that civil engineer will say, how much cement, how much steel, how many people should work for how many days? The civil engineer will give you a rough idea. Now I tell you, I burst a cracker in my house, or a nuclear bomb. Its yield was 10 to the power 18 Earths, or something like that. Was it a bomb or a mosquito yawning or something like that? Do you know? But you could say that elephants cannot sit in a car. Right? Nobel was given to proton pumping. These kind of things. Why did they goof up so bad? Proton is 0.0002 angstroms. Very small. Why? So like this, here
some cons, the structure is very similar, but certain things, just because of correlations and observations, they give. If people had gotten some concepts of physics very core engrained, okay, then that would not have happened. Okay, now you touch the wooden part on your table, all right? Okay, now touch the metallic part, right? Which one is at a lower temperature? Metal, right? 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 All of you agree? All of you are wrong. Wooden part.
Both are the same temperature.
Exactly. So there is very simple idea that both are at the same time. So here is conceptual understanding of very simple things. So if they had known, they know the definition, they will study, but they will not register. They will not try to translate the day-to-day experience like what you said. Elephants cannot sit in a car because it's big. How do you know it is big? Because we see it. Yes, that is your day-to-day life experience. This delta G, that, somehow that doesn't translate to your day-to-day experience. A civil engineer gets a hands-on feel about how many sacks of cement and all that. As a biochemist, as a biotechnologist, you are supposed to understand the time, the kind of the space idea, all that are important. And there's the physics part of it also is the. So when Danny puts something like this, which is being taught and which is being applied and which is being perceived as true the world over, it was something that even a child should have called out if he knew the basic definition of what an enzyme. Yes or no? Do you agree or not? Yes or no? Yes. So that is what we did. Have you heard? Now I give it back to Danny. Have you heard of Emperor's New Clothes?
Yes.
Hans Christian Andersen's story. I'll tell you that story. There's a story that you all would have studied. There was an emperor who was very fond of crazy things. He was indulgent in his own fanciful activities. He never paid much attention on the governance, and he was more interested in clothes and things like that, right? So there came around a couple of weavers, and they gave him a promise that they'll make him wonderful, fine clothes. You have heard this story? And then they say only the most smartest, the most learned, the connoisseurs of clothes, or the most finest people can understand the nature and texture of that cloth. I know I'm taking time, but this is very important for you to appreciate what Danny is saying. So you people see delta G equations and you shut your. This is way beyond our reach. No, it is not. I'm basically a biologist. I had no clue about these things. Just got some fundamentals clear like this. What is temperature? What is that is enough. So believe me, you can do this. So these people told these weavers, said to them that only the finest thinkers and seers and connoisseurs, they can understand. So what happened is, they took a lot of money from the emperor and they started enjoying it. And then after a few days, years went by, the emperor sent somebody to have a look. So when they came, they saw these people started to put on a show. Oh, see, going around, the yarn is there, nothing was there. So there was some action they were doing. So this man, being not so confident, he could not call out the bluff. He said, when he came back, because the work is going on. He just said, the work is going on. Yeah. And then, after a few days, the next in higher command went. So he had already said, they were already, and he also had some ego problems, and he could not also call it out. He also said. So finally, like this, a whole bunch of people go verify. Everybody sees because of some nonsense that people have already agreed upon. And finally, the day comes when they say, Raja says, I have had enough, I want to see what the cloth is. So they come, they do all this pretense, they put the cloth on him, and then the king goes out. Suddenly a kid comes and says, hey Rajananga. Basically, he had nothing on. So you have read that. You have come to. You haven't heard story? Yeah, emperor's new clothes. It's in all kids' textbooks. So the kid can call out not because the kid is smart. Kid can call out because kid has some basic funda. It's not bound by the social hoodwinks and all that. So that's what we are not very smart people. We know what the definition of enzyme is. If we knew which one is colder, the metallic one or the wooden one, if I had asked that, if you had the foundation of what the definition of temperature or heat is, or if I ask you which one has more heat, a cauldron of warm rice or a cup of hot coffee, can you answer? Both are same. Cup of hot coffee, cup of cheers. Exactly. So now you need to understand what heat is, what temperature is, what heat is dependent upon, what is temperature dependent upon, things like that. Here, coming back to this, calling out something as wrong, whether in textbooks or whether occurred by your teacher or whether in a symposium, all that can occur only if you have your attention here and you have some amount of fundamental awareness. If you just believe in parroting what has been taught to you, doesn't work. Did you understand? So, I took this 10-15 minutes extra to barge into his talk, to disrupt his talk, because a lot of things had gone. Was I right in telling you that you had lost this delta G and all that? Did you understand that? Yeah? Was it getting your attention, what you were saying? This part, did you see? Did you understand why it is? I think it's time for you to leave. No. This is very important. Did you understand? What happened? Huh? No, because this is very important. Otherwise, whatever Danny is saying, it is meaningless. We are not communicating. Here is the theory that LDHA in muscle and LDHB, they catalyze different reactions when they are isozymes. Yeah, basically structure is same. How will you have that? They had it. Why? Because of something, cause consequence, they just correlated without really verifying the basic aspects of theoretical foundations of enzymes. Yeah. Go ahead, Daniel. Sorry. Did you understand now? Yes. The LDHA, LDHB is very important.
Okay. So we had just a summary. We have a huge table there. I don't want to show that. It cannot be visible also. But all of these compounds, some of them are synthetic, most of them are natural. They also say that some of them are specifically inhibiting LDHA. Okay, in literature you can find that some compounds are known to be specifically able to inhibit only LDHA, which I found was weird because the active site of both these is similar, the structures are similar, the topography is similar. So we say I and O. I means inside, it bound inside when we did the docking. O means it bound outside. We did blind docks for several LDH A and B. You can see pig B, rabbit A, rat A, pig A. So what we wanted to find out is through blind docking, you don't tell the ligand where to bind. You just put a box around the whole protein. Most of them favorably bound outside. Also, some of them bound inside. Okay, that's fine. So you can see here that NADH, if you see NADH is supposed to bind to which one very favorably? Will be to A, right? NAD plus should bind to B, but both of them are okay, they are able to bind. Sometimes the orientation was ulta. Instead of the nicotinamide ring going in, the other part is going in, right? So NAD plus, for example, oriented in the unfavorable way, that is the ulta, in one of the dockings. It's just some visual thing for you to understand. You know, in the crystal structure very clearly of A and B, you got the coenzyme binding all the way inside, and then beyond that you got the negatively charged lactate or pyruvate being stabilized by two arginine residues. I had shown you that. So all of these inhibitors, I don't want you to read anything there. The visual here is just that on the surface you can see at some point some of the inhibitors are binding there more favorably. But yet they are known to be inhibitors of lactate dehydrogenase. How? Why? Is a question that we asked. Now, because these molecules have antioxidant properties, could it be that they are scavenging ROS also? Is that a mechanism, right? Some of them bound at the mouth, like crypt blocker. What you say is crypt blocker, that is entry of substrates and exit of product cannot happen like that. But in most cases we saw that there was favorable binding of even lactate to one part on the monomer surface. So here you can see that large molecules which cannot even enter the active site, for example, you can see PGG, something like that, galloyl. It's a very huge molecule. It doesn't go in. It cannot go in. But still it inhibits the enzyme. How? Right? So there is some other mechanism. What could that be? Also, if you see the structures of various mammalian LDH, yeah, the crystal structures, yeah, you can see here, for example, rat LDH. There is no active site separate. You can see that it's completely exposed to the solvent. You can see that in rabbit also, but if you see the pig enzyme, you can see that it's closed both A and B, right? You can see that there is a kind of channel which you can see, which I put a red circle. That is the alternate binding site which you are seeing in the yellow box, right? So it's not binding exactly to the active site in our docking. So we also have such diversity in these structures. In one of the papers I saw, they are saying that the rate limiting step of the enzyme is not hydride transfer. Hydride is direct 2 electron transfer from NADH to the pyruvate, right? It's not a hydride transfer that is the rate limiting step, but it is the flipping of a particular loop which is the rate limiting step, which does not make much sense, right? How can the movement of a particular protein loop influence the reaction or its rate? I don't know. I mean, if you know. So this is what we have come to finally, right? You've seen this slide before, that there can be a particular redox coenzyme which can produce ROS. That ROS can be involved in the reaction. But with LDH, you know, we can say that for pyruvate to lactate conversion, because the equilibrium constant is very high and the energy is very favorable, you have a very high negative delta G. the reaction could happen through the active site, I mean the conventional acid-base catalysis hypothesis. But what about lactate to pyruvate? That is our major question. So we have proposed that, based on our docking, we know that even lactate can bind somewhere else other than the active site more favorably. We have seen that. So what we have said is that LDH, because of the negative charge, it could stabilize to superoxide, and also NADH can also bind, and you can have multiple things happening here in the reaction, like superoxide binding and then NADH being converted and H2O2 forming because of reaction of superoxide and NADH. NADH can give rise to superoxide. You can also have oxygen being converted to superoxide. There are so many things that can happen. ROS can bind also in the active site, and if the substrate is bound on the surface, ROS can attack lactate also. So there can be many things that can happen. So there was a proposal that we would rename the enzyme to pyruvate reductase rather than keeping it as lactate dehydrogenase, because it does not, as the name goes, it does not dehydrogenate lactate. It only works on pyruvate and it reduces pyruvate to lactate, so we can call it pyruvate reductase. So there are multiple routes in the murburn scheme that can be involved, and there are other things that can happen also. Like for example, there is singlet oxygen, which is a, in photochemistry and all, there is singlet oxygen, which is an activated form that can, react with NADH and then give ROS or H2O2. So these kind of things can happen, and that could be the reason. ROS could be the reason for lactate to pyruvate conversion. Now to kind of prove that, this is a kind of mechanism where we can say that in the murburn scheme, lactate could bind somewhere on the surface of the protein, not exactly to the active site, and still be converted into pyruvate. So we can see that lactate was bound there in our docking result. I have marked that. So now going forward, this, I don't know if you are able to see this. This is an experiment done by Abhinav and his PhD scholar. If you take LDH enzyme, there is both the DLDH as well as LLDH. DLDH is a bacterial LDH which catalyzes D-lactate. LLDH is the one that we have in our system which catalyzes L-lactate. So both with D-lactate, lactic acid dehydrogenase as well as LLDH, what happened was, if you take the system is B is buffer, P is pyruvate, and NADH. Here we are monitoring the consumption of NADH spectrophotometrically. You can see that the rate of NADH consumption, if you put LLDH, for example, you can see that NADH plus LLDH, the golden color bars are very high, right? So look at the rate per molar per second. You can see that it's like 1500. That's very high. With three different buffer systems, the scholar did the work. One is phosphate buffer, another one is acetate buffer, another one is citrate buffer. In all, you can see that with at least not DLDH but with LLDH, you can see that there is NADH consumption is very high. But if you see the reverse thing, lactate to pyruvate in the three different buffer systems, the rate is much lesser. So the formation of LDH, the production of LDH is not as good as the LDH depletion, I would say. So then liver tissue was taken, and in the liver tissue you can see that there are different setups. You can see the colors. Both L and D LDH were added, but there is more consumption of NADH in tissue.
Can you just go back to the slide, please? Then?
This one.
Yeah. See, I have a suggestion. In the next time when you present this, you present these two simultaneously in the same scale. Only then this will fall, because otherwise you are magnified. There is also, they don't see the rate of the...
No, that's why I explained that.
No, that's not good. Presentation, you always watch out this. They will not see the axis. Yeah, because this is in hundreds and that's in thousands. That you don't present it like this, because
no, that is a raw thing.
No, but since you are visiting your class like this, no, I'm just saying. But you said it, but
yes.
Did you understand what was said? Everybody has got the switch?
Yes. They are completely out.
Yes, yeah. I know this is going to be hectic, but it's hectic.
3, 4, 5 minutes I'll finish. Okay, so what is according to the calculations which was done by Sir, this enzyme actually eats up NADH rather than making it, right? Do you agree from the previous slide that it more readily consumes NADH rather than makes it? Well. So if you take liver tissue again, now we are tracking that lactate to pyruvate. You can see that 3 different days the experiment was done. Day 1, day 2, day 3 doesn't mean liver was stored and no, not like that. 3 different days, but the trend is similar, right? You can see that you can see that the same trend of NADH is there. Right? So there is something called Fenton reaction. If you take hydrogen peroxide and you put iron 2+, you will get iron 3+, and you will get radical, OH hydroxyl radical. So he tried to see if Fenton reaction can give rise to high NADH formation. Right? Here we are looking at NADH formation. So the Fenton reaction you can see at pH 9. This is an important point which I should tell you. Lactate dehydrogenase assays have been carried out in clinical serology. They take unnatural, things like lithium lactate. Many things are very, very rich antioxidants and redox active molecules. They are added. Pyruvate to lactate is happening without any problem. Lactate to pyruvate, for that they are adding extra things which are non-physiological. That's a key problem which we have pointed out in that paper. So here, just Fenton reaction, when you have hydrogen peroxide breaking down into radical, it is able to give you a lot of NADH, and you can see pH 9 is very favorable. They are carrying out lactate to pyruvate reactions at very high pH in diagnostic setups. And they also tried adding superoxide, which is a very reactive form of oxygen. And when you add superoxide into the medium, you can see that only KO2 is also given. But the moment you add LDH, the NADH goes down, right? Repeatable. So with this, what I would say is they also did some FTIR to see if, you can see the trace for pyruvate, that is the red color, you can see the trace for lactate. The moment you add NAD plus and KO2, you can see that you are getting lactate to pyruvate conversion. Lactate is becoming pyruvate. So it was also traced with the help of GCMS, and the moment you have KO2 plus LDH, you can see that the amount of lactic acid has decreased. Right? With this, I don't know how much you have understood. You are very tired. This is the end of the day. Thank you for your patient listening. If there are questions and I can answer, I will definitely do.
Please put your hands together for that. One thing, when we published this paper, we just did docking only. Something that anybody can do here, right? If you had ordered off, we do. So, one thing Danny did not stress is, Danny, can you go back to that slide? I know you're busy, but see something that very simply you can, certain things, go back. This is wonderful data. Go back. You did not stress on this part. What did you see? You said some ulta. Where it happens ulta is what you need to see. The NAD plus, it is supposed to get reduced to become NADH in the reverse, the reaction that is physiologically infeasible. Do you think it binds? If the electrons are coming from the active site is here, right? And the lactate or pyruvate in the classical should be inside. So which one is the nicotinamide moiety? Nicotinamide moiety is which one? And this is the base and then the nicotinamide, right? So the positively charged nicotinamide will not go in. That's what is shown here. Common sense, you can get NAD plus will not go in and orient with the proper way like that. That's what Danny has shown here.
Because the arginines are
there. Right? Positively charged arginines are there. So if the theory, go back slide, that if it is going by the same route, that pentagon.
Which one?
We did not
that pentagon.
The cancer thing, but
here.
they have had enough. You see here, the classical theory says this way. So the binding of everything is in the same, they say, but they say this has to go in, then that has to come, it has to act at the active site, that way in both. How can it be? The docking clearly says it cannot dock like that. So very clear, and common sense also appeals. Positively charged NAD plus, multiple arginines there. It will not, it will repel. So that's why you get dock like that. So what common sense predicts, what simple things predict, these scientists, because of the pressure of norm, you understand, they have conformed. I told you, 1 by 1 foot box, they put some cone in and try to put extra. Whatever is not accommodatable, accommodate. Don't become scientists like that. Alright? So, yeah, please ask questions to them. I know.
They are really done.
They are really done.
Or completely not understood.
Yes, you have been sitting from 10:30.
I just wanted to add.
Yes, sir.
Like Sir has been constantly saying that, scientists, they will, because of some peer pressure, they'll put up some theory. I have seen this happening. Okay. Like, I was in a lab where the mentors believe that gold nanoparticles have some fluorescence. They don't. Okay.
Gold
nanoparticles, they do not have fluorescence property. Fluorescence property they don't have. But then somebody in China had reported that. And then because of that pressure, somebody in the lab also agreed to it. That was a false data that she observed, and she reported that to be fluorescence. And it continued to be in the lab like that for a very long time, unless it's broken. So this truly happens, that because of some pressure also, scientists will push in a theory and it will propagate for years and years together. So, question.
Don't accept. whatever you don't see logically, simply. I tell you, we are not intellectual geniuses and all that. Basic foundations, basic aspects, don't get scared. Whatever you don't understand, you say you don't understand. I told you the story of Narasimhan sir, Krishnadas, don't forget. It's very important. You might not, whatever these structures, all these details that we are presenting, not important. What is important is the story that is coming with that. Have the guts to call out, and if you'll not have guts if you start talking, dissipating like this. This kind of meetings, you are young. We never got a chance to attend any symposium at your age. We never. I'm looking at you people only because, when young, you can have ideas. You can have, by the time you get a little older, the system would have crushed it. Do you understand? It's very difficult to break that system, that psyche. It's very, very difficult. Only if you're really fortunate, you get some exposure, some people in your lives who urge that kind of mindset. Otherwise, it's very difficult. All right. So let's just say someone 7:30, we'll let you go. Mudinjiruchu. So I think just some interactions now. What was the take-home story for?
Hello.
So this is what it has to be about. It is about empowering our younger generation the right guts to approach scientific interaction and discourse. Yes, that's what we are here for. Great. That's why you people were given a chance. I know. See, I was joking with him. Ultimately, can I be honest with you? Can I be? It's very cruel. Most of the people won't understand. Even if they understand, they won't care. Even if they understood and they care, they can't do a thing.
Yeah, not to,
Yes or true? Yes or no?
Not in this.
Yes, but yet we are doing it. Why?
You get by attending this.
Why are we doing it? I know you people are way beyond your. It's gone. You want to go out, drink a cool drink, put something in your tummy and go. What that body was subjecting us to, man. You want to say that, right? I get 3 days with you. We'll work on it. So, what was it?
Yeah, you asked.
We'll come back. No,
So we can't
this is
determine.
torture. Pure torture. I enjoy being a dungeon master. Come on. Go ahead.
So we can't determine the particular structure.
Is this what you came for? Pesra.
So we can't determine the hemoglobin structures particular manner.
No, don't make me do all these stunts, man.
So we can't determine the particular structure of hemoglobin. It may be slightly change different blood groups also. Like for positive or negative blood groups, it will slightly change, and the hemoglobin levels also the scalable is low and high. How you determine this manner in common way?
Let me say, his sound is good, but the question articulation is, I don't know. Positive and negative. What is positive and negative in this context? RH factor. RH positive. Okay. What is RH factor? It is an antigen on the cell surface. So he has studied something. Now he is asking something. Is coming out. All right. That's good. But நாங்கெல்லாம் கபடி ஆடுறோம் நீ கோகோ ஆடுற அந்த மாதிரி ஆயிடுச்சு. No problem. No problem. I'll tell you as it is. Pardon? Pie chart. That was not a pie chart.
Graph.
Which graph? Now you understand. So
Hemoglobin graph.
You come out, you might not be articulating well, you might not get there, but this is one step at a time. We are not getting there. I'll get you later, but then at least your sound got, and perhaps I'm not getting your articulation. Okay? So, good, we got something about hemoglobin. That is good. We got something about it. Good one, Janidhi. And what about lactate dehydrogenase? We have, for you people, let me tell you, Professor Yahya is with us. Please, sir, he is Calicut University chemistry head. Is traveled all that far just to be with us. There are still people who value good education, good scientific discourse. Otherwise, he need not have come. His student was leaving to Sweden today. So I wanted to introduce him as soon as he came, but then he said, we'll talk after that. But then I tell you that there are still people out there who will help you if you show the right attitude. There are people who really care. There are. And then there is a system that is, because what happens is many people don't interact and ask questions, and so the system slowly shuts itself down. So you need to shake yourselves up a bit. LDH. And then, all right? Okay. LDH, what did you get? No, don't, no, this lateral, no, it pains. No lateral. See, you are also doing it. They are also doing. Oh, so he's talking, sorry. Anybody? LDH? That confidence, when somebody is asking you, it is your time to speak. If you do this, that means you're not taking up your opportunity in time. Did you understand that? How sad it is? You have the opportunity to spar with me, to interact with me. You've come this far. Why did you come? To sulk away? No. You have come to interact. So what is it that you learned about LDH? So, yes, LDH. Most interesting thing about LDH. Huh? Pardon? See this, sir, whack. What is the most important thing you learnt about LDH from Danny? Let us refresh. I'm not letting you go that easily. So see this, and then I'll ask you the question again. All right. So I just gave you a refreshing. That the other slides were prepared by Danny. This was prepared by me. What was the difference between Danny's slides and mine? I'm going to be critical about Danny right on his face. Abhinav told him not to do that, but still he did it, so he has to get it right. We do business like that. Yes, what was it? Huh? Letters are one, partly right, but it was like a story I'll tell you. One guy took a frog. All right, he was a biologist. So this is the approach towards accuracy and precision and all that. So he's a biologist. I'm very critical about biology. So he took a frog and then cut. First day he said jump. The frog jumped about 2 feet. Jump 3 feet. So he took many frogs like that and subjected them to the same condition. Every day he'll do some bunch of experiments. He'll make a table and then he'll say, the next day he got another bunch of frogs. He said jump. Before saying that cue, he cut one leg off. So the next, then he would say jump. It will just move. Then the jump, move a little bit more. So like that, second day he cut 2 legs off and said jump, jump again and all that. So third day he cut another leg and then finally he made a table, and final interpretation of the data was, if you take a frog on the third day, if you cut off 2 legs, it doesn't hear properly. Because on the second day, when he cut 2 legs and said jump, jump, there was no real difference. One good interpretation. The fonts are small, big. Okay, that's not getting the main story. It's not wrong. The precision somewhere okay, but accuracy no. We have to get to be accurate also. Yes. It's 8 years problem. I've been waiting for students for 8 years. So I will not let you go that easily today. So tell me. Tell me. Huh? See, you want to communicate, but you don't want to be heard and responded to. No. And when I say yes, that soul goes back into her mouth like that. Boom. It has to change. Yes, mic. Mic for you. No mic for you. Come on. Yeah. வெறும் காத்து தானுங்க வருதா? ரேவதி சொன்னால தேவர் மகன் அந்த மாதிரியா? இல்ல just it's necessary. Speak. What's your name?
Akshada.
Akshara.
No, Akshata.
Akshata. Good. Akshata. Right. I heard that, but I heard it was unfamiliar. Good. Don't look at her. I told you I'm the beautiful guy. Go into yourself. Oh no, she's dead. Okay. That's what, to face the world and say, That's wrong. If you are going to be sitting, sulking away, you are shy. Will you be able to do it? No. This is the first step. Start interacting with seniors. Ask questions in the class. Don't chat away in the class. You keep chatting away amongst yourself. That's the reason you are doing it. And stop doing that. Try to practice. Alright? Alright. Go ahead. Tell me, anybody who learned something about LDH. These two people, poor guys, they are not getting, usually resource persons get some money and all, right? I'm not going to give them a damn thing. They just come. Yesterday, one of these guys slept on the floor, poor guy. He said that he had a shoulder problem, so he got the bed. So I'm telling you, somebody has to tell me something about LDH that they learned. Otherwise, you're not going anywhere. You're not getting. Yes, not you. These are BTech kids. Any BTech kids? Yes, Danny. Yes. BTech kai Not BTech kid. He raises his hand. Once again, that accuracy, precision gone for toss. Yes. All vague comments not addressing the question. The question is very clear. We are playing a game. The question is very clear. What is the question? Something about LDH that you learned from Danny. Otherwise, Danny, that is precisely what I said. What was wrong with Danny's PowerPoint presentation? Too much information. Too little time. So we need to be focused. That's what. So, but Danny is very passionate. He is very meticulous in what he does. So, in this time, he wanted to give a lot of information to you. And he will be very unhappy if you say that you didn't get anything. Alright. So, BTechs, can I say that BTechs are... Somebody wants to salvage BTech pride? Yes, you want to? Say something. One second. No. That is not.
LDHs.
Yes.
LDHs are accumulated in the muscles, sir. In heart muscles, LDHB is accumulated because it overpumps. It doesn't stop its working, so LDHB is accumulated on heart.
Okay, but that is a classical view that LDHB is in the heart, so it pumps, and there is no lactate, so you don't have the muscle cramping. So that is a classical view. That is, I said that is not. No, that is not right. Right? I told you a story about elephants from Kerala, Kunjiraman, Appukuttan, all that. Remember that? So now we have another problem. Yes? Anybody? BTech? Nobody will give you this opportunity. You will not even be looked at. In our school, we train our BTechs with lot of opportunities like this. Yes. So, yes, Niranjana. I know your name already. See, I know Niranjana's name already. Why? Yes. Real people will value real attention and real purpose. One question she answered, and that's why I remember her name properly. And even if you don't answer properly, if you're interactive, I'll remember you. I'll forget you.
Thank you, sir. One thing I understood was LDH is considered as a cyclic enzyme as it converts pyruvate to lactate and lactate to pyruvate. But sir said that it doesn't do the inverse reaction.
Wonderful! One of the most... Put your hands together for her. That's one of the paradigm shifting ideas that murburn concept ushers. That reactions with large difference in delta G, that is, free energy of reaction, those cannot work from, I told you, what was the example I told you? I showed something. Jumping. I told you that I can jump from here to here and there to here, but I cannot jump from here to Himalayas top Everest top, and I cannot jump. I might jump back from there, but I cannot jump again. Right? Yeah. So you got it right. So good. So that's very good, Niranjana. What else? What else? It's only a few more minutes and done. And we'll pack off. Done, done, done. See, I think you take him. He is very conversant with you. He's not talking to us. He's not interacting. He should interact with us, no? Yeah, that is good. Anybody else? So I think we'll come to these people. Yes, my dear seniors. You're the people who are doing some research, right? Yes, Mike. So what is it that you think was interesting about the LDH that Danny presented?
What I think was interesting is there is a real gap between the delta G values, because the reverse reaction from lactate to pyruvate, it's taking plus 476 plus. So it is not that easy for enzyme to get over that much of interaction energy and go down. And simply in the visual point of view, no way that the positively charged nicotinamide ring is going to enter into the active site.
Basic things. Theory doesn't match. Experimental data doesn't match. Danny also said for the experimental assay for the reverse, they use unreal conditions, right? So unreal conditions, if you try to do, you can say you put a lion in a circus ring and then put a flaming loop and make the lion jump through that loop. And then you put a hypothesis. We showed a lion jumping through a flaming loop. In jungle, lions jump flaming loops and then they get rewarded for it. No? Is that a good theory? No. We can show whatever we want. We are capable of that. But how it is in real place, that's what counts. What's the time now? 8. I think that's fantastic for first day. Tomorrow, you will get food only if you attend the whole symposium. We are spending how much money, you know? You have any idea? 200 rupees. What is that? Can you get anything? We are paying you for stay, khana. No, bata nahi raha hu. I'm not trying to drive it into you. I'm just trying to show you that we are great. I'm just kidding. I'm just... दूसरे तरफ से भी, you know.
So the hostels are actually great.
Yeah. So good. No, I'm just trying to be funny here in the sense, heads I win, tails you lose. The point is, please. Tomorrow, will you be more interactive? Wonderful. Because tomorrow, the system is more complex. We are coming to, I put easier system, simpler system. Tomorrow is drug metabolism, $1 trillion industry. And then respiration, heat production, photosynthesis, electrophysiology, vision. It's going to be packed and the systems are much more complicated. But I hope, thank you so much. Welcoming Professor Yahya again. Our Dean sir is here. Sir, do you want to say something? Yes, if you want to say that. There was this wonderful AR/VR thingy that we had put here. Did any of you see it? How was it? Yeah, 3-dimensional ideas, no? Yeah, but always think in terms of, if you're a biologist, think in terms of real space, real time, real forces, real. Get it real. Just don't buy anything like this. We'll go for dinner. All right? Thank you so much. Come with me, otherwise you won't get food, because I've directed them, don't give food. Sir, there were so many people here. This is the state. How can we give food to them?
Hello. Prabha, Yashpal. Yashpal. Sorry. Yashpal, hello. Yashpal. Yes, Yashpal. What is your question? Yashpal, you are not coming clear. Yashpal is a assistant. Oh, is offline. Chalo. Okay then. Bye. Yeah, sir.
Yashpal, please ask your question.
Please ask your question, Yashpal. He has been from the morning. Sorry, Yashpal. Yes, ask me. Wonderful question. So we are currently, Professor Prasad Burra, his line is not steady. That was a nice question. The question was, yeah, we are trying to get that. We are using some AI protocols and some basic, prompts from our side to try to make something. Hopefully by Thursday, we will be able to release the website, the basic form at least. We are trying hard. All right, we are at it. We are at it. Thank you. Wonderful question. So people are waiting for dinner. Thank you, Yashpal. Thank you so much. Bye bye. We'll see you tomorrow. So how come?
Bureau room bureau ఉంది సార్. డిన్నర్ అరేంజ్ ಮಾಡಿದ್ರು.
I told you an example of the oil.
முடிஞ்சு முடிச்சல கலங்க.
Sir, wait. One second. No, one second.